Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is part B of lecture 25. The topic is gene mutations. This topic will be discussed under the following headings. After watching and listening this lecture, the students will know about various types of gene mutations and how they affect the gene product. Introduction. Any change to the genetic information of a cell or virus is called a mutation. Genetic information is stored in DNA. Deoxyribonucleic acid is the genetic material of a cell. DNA is composed of nucleotides. So the changes which takes place in these nucleotides encompasses gene mutations. The word mutation was for the first time used by Hugo de Vries in 1907. Gene mutations are also called point mutations, and these are the chemical changes in just one nucleotide pair or few nucleotides of a gene. Mutations has another type which is chromosomal mutations. Chromosomal mutations are large-scale mutations. They affect the whole chromosome, whereas gene mutations are small-scale mutations and usually affect a single gene. But a single gene, if holds an important position in the body, will have drastic effects on the organism if mutated. Here is the central dogma of life. This was explained by Francis Crick in 1956. You know Francis Crick, he was the co-discoverer of the structure of DNA. The central dogma of life means that the transfer of genetic information in a biological system occurs through this pathway. The DNA carry instructions for the synthesis of proteins, enzymes, and for the development of different traits. And proteins are the entities which usually carry out these functions. And DNA instructs the synthesis of proteins via an intermediate which is called RNA. On the right side of your screen you can see that a double-stranded DNA acts as a template for the synthesis of messenger RNA through a process of transcription, and then this messenger RNA along with ribosomes guides the synthesis of a protein. Here is the genetic code. Genetic code is the set of instructions which guides the synthesis of a polypeptide chain. Sequence of nucleotides in DNA and RNA that determines the amino acid sequence of proteins. The genetic code consists of 64 codons. 3 of them are stop codons, which signals the termination of translation, whereas the other 61 are protein amino acid encoding codons. How we came to know that gene mutations causes genetic disorders. It was courtesy of the work done by British physician Archibald Gerard in 1907. He wrote a book, Inborn Errors of Metabolism. In that book he stated that gene mutations are responsible for defective enzymes and proteins. He was studying a genetic disorder, alkaptonuria, in which one of the enzymes is missing, and the broader problems occurs in the metabolic pathway, and the person suffers some serious consequences. So he said that these defective proteins or enzymes are because of defects in the gene from where they are synthesized. Gene mutations are only transferred to the next generation if they take place in the germ cells or gametes. Genetic disorders or hereditary disorders are because of gene mutations. Here we see an example how gene mutations affect an individual. Our RBCs or red blood cells are composed of 4 polypeptide chains. 2 of them are alpha and the other 2 are beta. So here on the right side of the picture you can see that mutation in one of the beta globin genes results in an altered amino acid valine, and as a result, if such defective polypeptide chain becomes part of RBC, then the RBC becomes sickle-shaped. The sickle-shaped RBC is unable to carry oxygen properly. Moreover, it is very fragile and usually broken while passing through narrow blood capillaries, and the person suffers from anemia. So here you can see that a single change in the nucleotide sequence have drastic consequences on the organism. Majority of the mutations are harmful. They decreases the fitness of the organism. However, there are certain mutations which are useful. In fact, mutations are one of the mechanisms of evolution, and new alleles only arises through mutations. You know, the genes exist in pairs, and the individual members of the pairs are known as alleles. So new alleles in a population only arises through mutations. Classification of gene mutations. Gene mutations are broadly categorized into 2 types: base substitution. Base substitution means when one nitrogenous base is replaced with another nitrogenous base. There is no net loss or gain of nucleotide. Only replacement of nucleotide takes place. Base substitution is further divided into 2 types: transition and transversion. We know that DNA is composed of 4 types of nitrogenous bases: adenine, guanine, thymine, and cytosine. Adenine and guanine are double-ringed large bases and they are purines, whereas thymine and cytosine are single-ringed structures. They are smaller bases and they are pyrimidines. So, if a purine base in DNA is replaced with a purine base, or a pyrimidine base is replaced with a pyrimidine base, such type of mutation will be termed as transition. Transversion occurs when a base of one chemical category is replaced with a base of a different chemical category. A double-ringed purine is replaced with a single-ringed pyrimidine, or vice versa. The second major type of gene mutations is base insertion or deletion. Here, a loss or a gain of a base takes place. Either a completely new base pair is inserted into DNA or a base pair is lost. So, insertion and deletion occurs in this type. Therefore, these mutations are also known as indel mutations. Here are the molecular consequences of gene mutations. Means if mutation occurs in a gene, what will be the effect of that mutation on the organism carrying those mutations and on the instructions which this gene has. We will discuss each of them one by one. Here is a general overview of various types of gene mutations. On the right, you can see nucleotide pair substitution, and on the left, there is nucleotide pair insertion or deletion. At the top tier, you can see a double-stranded DNA, the strand which acts as a template is the one from which the messenger RNA is copied. In the slides, we will discuss each one of them in detail. So the first one is silent mutations. Silent mutations are usually as a result of base substitution. You already saw the genetic code and you know about codons, which are the triplet of bases, and they represent amino acids. So in this type of mutation, one base is replaced with another base, and the new triplet codon which is formed luckily represents the same amino acid which was represented by the original codon. And what is the reason for that? Why this happens? Because the genetic code has 61 amino acid encoding codons and 20 amino acids. So there are certain amino acids which are represented by more than one codons. So this mutation is a blessing in disguise, where despite alteration in DNA sequence, the organisms doesn't suffer any consequences. And these type of mutations are also known as synonymous mutations. And here, although change occurs in the codon, but the codon represents the same amino acid which was represented by the original codon. So there will be no effect of such mutations on the organism. Missense mutations. In these type of mutations, a base substitution occurs in a codon and the codon is altered, and now this codon represents a different amino acid than the one which was represented by the original unmutated codon. So these mutations are called missense mutations or non-synonymous mutations. Nonsense mutation is the most drastic form of base substitution. Here, change occurs in a triplet codon where one of the bases is replaced with another base, and the new triplet codon, the new triplet combination formed represents a stop codon. The original codon represented an amino acid, whereas the newly mutated codon represents a stop codon. So this type of mutation will create a stop codon right in the middle of a gene, and there will be premature termination of translation, and a defective, incomplete, abnormal protein will be formed. Now we come to the molecular consequences of indel mutations. As you already know, indel mutations are insertion or deletion. There is a net gain or loss of a nucleotide, and these mutations usually result in frame shift mutations. What are frame shift mutations? You already know that During translation, messenger RNA is read as triplet of codons, means 3 nucleotides represent a codon. So in this type of mutations, a base is inserted or deleted and a new combination of codons is formed from the point of insertion or deletion. There will be a new combination. The whole reading frame will be disturbed from the point of insertion or deletion of that particular nucleotide. So complete change will take place, and the new triplets formed will either represent an amino acid or not. And the frame shift mutations are usually affecting triplets of bases, but if the inserted or the deleted nucleotides are divisible by 3, then there will be no change in the reading frame beyond the point of deletion or insertion. Here you can see that 3 nucleotide pairs deleted and no change occurred in the codons beyond the point of insertion or deletion. Mutations are caused by chemical and physical agents which are called mutagens. Now the mutations which are caused by mutagens are called induced mutations. There are certain types of mutations which are spontaneous in nature, means they occur on their own inside the cells because of the processes which takes place inside the cells, like replication of DNA, recombination, that is crossing over, or repair. And that was the end of the lecture. Thank you.